Yeah, I'm gonna talk about biochemical-based newborn screening using dried blood spots. We're gonna be using liquid chromatography tandem mass spec, LC, MS. Measure enzymatic activities and biomarkers in the same go. And I think it's safe to say there's no other technique that can come close to the speed and degree of multiplexing that we can do with LC, MS, as you'll see. It's now in routine use in several newborn screening labs worldwide. I mean, a lot of people are using mass spec based for LSD, but a subset are using LC mass spec, Illinois, Utah, Connecticut, Taiwan. Other labs are now using a lower multiplexible technique called flow injection mass spec, but are now upgrading to LC. I think when we talk about MLD newborn screening, they will have to do LC. So I think it's just a matter of time before LC replaces flow injection. I think it's proven to be robust and high throughput. There's a lot of bad rumors floating around about upgrading to LC is difficult. It's just not true. We've run pilot studies, several out to 100,000. Illinois is at 500,000 or something. Taiwan is same level. So it really doesn't add any complexity to the system. I think that's that. Now, don't get scared. This is our current 27 plex. So it includes many things that are treatable and some that might become treatable in the near future. We cycle between positive mode and negative mode. Some analytes are detected in positive mode, some are detected in negative mode. For example, CTX, we detect a bile acid, and for MLD, we detect sulfatides. This can be done at about 2.1 minutes per newborn, so it's a little bit slower than flow injection, but it gets you so many diseases. To be honest, if you look at the several hundred diseases that are now being considered for DNA sequencing panels for actionable pediatric diseases, we're getting close with this assay to cover everything that I can imagine a biochemical screen on the horizon, right? So most of these diseases that will be in DNA panels are things that's difficult to imagine a biochemical biomarker in a dried blood spot. But with LC mass spec, I think we're getting close to covering all of the things that I can imagine doing with blood spot analysis with biomarkers. So now let's focus in on MLD and CTX. So we did the first pilot study in 2020. We did 28,000 de-identified newborns, measuring the sulfatides for MLD and the bile alcohol for CTX. For MLD, we measured elevated sulfatide. And then when that was high, about 0.7% of the time, so we have a high reflex rate, you absolutely need the second tier ARSA enzymatic activity done in the same blood spot. There are reasons why we don't do the enzyme first. It's a little bit harder. And then for the third tier, we did DNA. So we had 200 elevated sulfatides out of 28,000 is about 0.7% that went to ARSA. All of them had more than 30% of normal ARSA, except one had 8% of normal and DNA showed a false positive. One had 0%. And genotype is a proven case of MLD. This is a genotype that we find often in the literature with clinically early onset MLD. So we have 0 to 1 false positives and no false negatives out of 28,000. How do you know we don't have any false negatives? That's a story for another day. But let's say we tested over 20 newborn blood spots from kids that were diagnosed with MLD, and we don't miss any of them. So that's about as good as evidence as you can get that we should expect very few false negatives. I think newborn screening doesn't get any better than this. I mean, to be honest, I think Krabbe newborn screening with cyclosine is this good. And I think MPS 1 and MPS 2 newborn screening with GAG analysis second tier is also this good. So it's clear we need these second tier tests. And then we get to levels of precision that are pretty much unprecedented in newborn screening. Really nothing works better than this. With CTX, we measure a bile alcohol intermediate we call tetrol G. We had only one elevated hit out of 28,000 DNA proved a clear genotype for CTX. False positive rate is 0, false negative rate is 0 out of 28,000. Okay, so we're pretty happy about this. It's all published. Now, Fred Vaz and Hedy Hudekuper in Amsterdam, they're the ones that discovered the tetrol G marker for CTX. So they recently finished a pilot in Amsterdam on 20,000 de-identified newborns. They did both flow injection and LC. They both work. The LC is a little cleaner, but they both had no false positives. And they did not detect any CTX in this case. We got 1 out of 28,000. We got a little lucky. We think the incidence of CTX is something like 1 in 100,000, but we don't know for sure. So it's kind of good that CTX can be done by both flow injection and LC, but I think it would make more sense to do it by LC. It would be for reasons I won't get into. When you do CTX, you have to be in negative mode, and when you're doing it with acylcarnitines and amino acids in positive mode, and then you have another 30, 40 seconds of negative mode, it's going to double the amount of time you need for flow injection. You don't get it for free. It's not like ALD. The biomarker is very low abundant. You need, almost a minute of data collection to see the biomarker. So by flow injection. So I think it makes more sense to do it by LC, but you have a choice. Now I'm happy. So about a year ago, David Casper started a company called Archimед Life in Germany, and he went live, not pilot, live for newborn screening of MLD in hospitals that cover about 20% of Germany, and he'll start soon in Austria. Out of the first 100,000 in Germany, they have identified 3 cases. 2 have a genotype of juvenile MLD and are being offered this new gene therapy, LibMLD, which is approved in Europe and England. And then they had 1 genotype, the juvenile adult boundary, and would be off label for this treatment. So will not be approved in Germany. So I don't know what the conversation looks like between the doctors and the family, but probably not good. The publication of this is pending. This is a live study. Now, in the meantime, we've had an explosion of interest in newborn screening for MLD. So I mentioned my study and then Germany, Austria. Screen Plus in New York that Joe Orsini, Melissa Wasserstein, Dieter, and I are involved in. They've screened about 12,000 we screened for 14 diseases, including MLD and CTX. The goal is 25 to 30K per year over 5 years. We're getting close to that goal now. And no hits yet. Florence, Giancarlo Lamarca started recently a pilot, 25,000 a year for MLD. Normandy, France, this is interesting. Soumaya Bekri, Normandy, France, this summer will start 100,000 over 3 years, MLD and about 7 other LSDs. Saudi Arabia has a small pilot study in a hospital in Riyadh that started. Illinois has a bill, it passed 106-0 in the state house, and funding was appropriated to go live with MLD as soon as the state lab can set up the test. So we're working with Kaja Bashir-Rudin to set up the MLD test. They are live. Manchester, England, Heather Brown did a pre-pilot study of about 5,000. And Tennessee is supposed to start a pilot study sometime at the end of this year. So you can see there's an explosion of interest for MLD. Now, as far as a RUSP nomination, we would use the German perspective pilot data as that'll settle that, I hope. We're waiting for the LibMLD to be approved by the FDA. That was the decision to make before we would do a RUSP nomination. I think the treatment seems to work really well, and we should wait for that. Been a little controversial, but that's what we're going to do. Yeah, I just said that. So for CTX, we don't have a prospective pilot data hit yet. We just have mine in this de-identified study, which doesn't count. We're hoping Screen Plus will identify a new patient in the coming years. I am at a total loss as to why the Secretary Advisory Committee requires a prospective pilot study, since we have already proven that we have a very good test for CTX, perfect. And treatment with bile acid supplementation is the standard of care and would be offered to pre-symptomatic newborns. So I continue to push the Secretary Advisory Committee to reconsider this so-called N of 1 rule. What point does it serve to find one more CTX patient? To this end, I am recruiting CTX families to attend an upcoming Secretary Advisory Committee meeting in Washington to show the importance of early diagnosis for this disease and the suffering that these people have endured, missing the chance for early treatment. Okay, that's my little rant. Now I want to change gears in the last 10 minutes or so, talk about something I think is pretty exciting, something new. Well, little introduction to that. So we want to improve the genotype phenotype for MLD. We have a new publication coming out this summer in Genome Biology. It's called Predicting the Disease Severity in MLD Using Protein Activity in a Patient Phenotype Matrix. It's a collaboration with my lab and workers at BioMarin over about 5 years. We collected all of the patient data sets from the literature and genotypes from the registry of the MLD Foundation. You know, not everything's published. That relates disease severity of MLD to the ARSA genotype in 488 individuals with diagnosed MLD. Yeah, the set of all ARSA variants found in late infantile, which is the most severe form, we define as severe variant. So any variants we find in a late infantile, we define as severe. Okay, that's a definition. For juvenile, we almost always see one severe as defined above, and we also see one not on the list of severe, which we therefore define as moderate. Okay, that makes sense. And then for adults, we find sometimes one severe from the list. The other we don't call moderate because that would have been juvenile. We call it mild for obvious reasons. And sometimes we have moderate cases. Adult MLD is very rare. It's sort of like the distribution is a little bit like Krabbe disease. Most of it's the early onset, more than half, and then the rest is, variable onset. So we've expressed all of these variants along with 300 VUs that we found in the NOMAD database. This is the largest human genome sequencing database in the world. It's 125,000 human genome exons. We expressed all of these, so all of the published variants and 300 VUs, variants of unknown significance, in 293 cells, and measured the ARSA enzymatic activity. What we found is that severe mutations give about 0 to 2% of residual ARSA. Moderates give 2 to 4, and milds give 4 to 13, and benigns greater than 13. This is how we bracketed it based on the observed data. This is all in the paper that will be published. All right. So then we take all the VUs and we measure their activity and we assign their bracket scores and tell you whether it's severe, moderate, and mild or benign. Then we take the phenotype matrix, which is, if you're severe, you'll have late infantile. If you're severe, moderate, you'll have juvenile, et cetera, to predict the worldwide burden of MLD and its stratification. This is the key observation I want to point out. One of the eye-opening observations is that 20% of the VUs in NOMAD are severe. So 20% of the VUs in NOMAD kill the enzyme activity. They're bad guys. Okay, come back to that. I want you to remember that. Now let's consider first-tier whole genome sequencing for MLD. So probably don't believe me, but we've recently done the pilot study on 125,000 human genome sequences. We've analyzed the ARSA and a bunch of other LSDs. We've actually done a pilot study on 125,000 humans at the whole genome level, first tier. Okay. And the data is already there. You just have to go inside the Broad Institute and get all the data. We've already done it. So we're talking about all these pilot studies. We've already done it. The paper is coming out in Genetics and Medicine Open. I can just tell you what's in 125,000 humans now that we've done the pilot study. 49 are VUVUs. If we extrapolate across the USA, 4 million babies, there'd be 1,500 VUVUs per year, okay? That would be found by newborn screening. Presumably, most of these would be false positives. But remember, 20% of the VUs we know are severe. That reduce the enzymatic activity of the protein to 0 to 2%. And so we would estimate with all of this together a false negative rate if we were to sequence for screen for MLD by first year DNA sequencing of about 50%. Okay, that's pretty high. It's already been shown. The pilot's done. The data is published. It's 50% false negative rate. That's your answer. Okay. So I'd like to say that first-year whole genome sequencing should not be used for newborn screening for diseases in which we have well-performing biochemical dry blood spot screens, for example, MLD and CTX. And especially for diseases in which we can easily add the biomarkers to an existing LC mass spec multiplex, i.e. MLD and CTX. We don't need more pilot studies to prove this. We already have all the data and it's published. We already have first-year biochemical newborn screening data on 28,000 babies in Washington and 100,000 in Germany. And we already have first-year whole genome sequencing on 125,000 humans. Okay. Yet, I'm a big fan of first-year newborn sequencing of diseases that are actionable and pediatric diseases in which a biochemical newborn screening method is not on the horizon. I'm actually a big fan of this. What about all the VUs? Okay, I showed you it's a big problem, but you have to change your mindset a little bit. So Genomics England will ignore all of them, all of the VUs. What about all the VUs? They're just going to ignore them. There's going to be a false positive, negative rate, 50% in the case of MLD. They will say it's better to screen for these few hundred actionable pediatric diseases and catch half of them or some of them rather than to do nothing. That's pretty reasonable. Hopefully the false negative problem will decrease over time. But I'm still very worried about the false positive problem because late onset variants are also American College of Medical Genetic pathogenic. So you see Genomic England says they will call out all the pathogenic, American College of Medical Genetics, pathogenic and likely pathogenic variants. But this includes all of the variants that are drivers of late onset disease. And we know when you have two late onset variants, you often don't get a disease or at worst an adult disease. You see this in MLD, you see it in Krabbe disease. And so we're gonna have a lot of false positives, okay? So they're gonna have to figure out a way to focus on the pathogenic variants that are the more severe. Okay, they're going to have to ignore the late onsets to some extent. Otherwise, they're going to have a massive false positive problem. And that is the #1 challenge facing newborn sequencing, in my mind. All right, how much time do I have? 5 minutes? I want to talk about something very exciting now. Voodoo. It's a new technique that we developed. We're going to do a lot of VUs. We call it voodoo. So in my lab, we practice voodoo, okay? And it's really cool. Massively parallel, inexpensive, and rapid biochemical protein folding annotation of every possible single site amino acid substitution for any cytosolic protein of interest. So we're talking about what does this mutation do? What does this mutation do? I'm going to tell you what it does in all possible mutations for any given protein without ever seeing them in patients. That's a pretty bold statement. Let me show you what we can do. It's something like that. I know it sounds ridiculous. Now remember, we think that most genetic diseases are caused by missense mutations that disrupt protein folding, leading to protein degradation. Certainly not all mutations, right? But most of them. We have a sense of this. For example, Wilson disease, a copper transporter. If you look at the amount of Wilson protein in cells from 150 confirmed Wilson patients, more than 90% of them have essentially no Wilson protein, okay? Likewise, if you measure lysosomal storage diseases by the absence of the protein, Hopwood showed that 90% of the time you catch them. So most of the time you're missing the protein. Most of the time you don't have an active site residue where the protein's folded and it's there, but it doesn't work. Yes, you do, but not most of the time, we think. So now we got to figure out what are all the missense mutations, the single site amino acid substitutions that wreck protein folding. And let's just do it for all of them. That's what we can do. So we obtain a library of DNase coding for the protein of interest where each amino acid is replaced by all 19 other options at 1 per protein. This can be easily done now for about $5,000. We transfect a single tube of 293 cells. The cells are engineered to ensure that only one variant is expressed per cell. Now, how we do that, I can show you later. Guaranteed, every cell expresses only one of these variants at a time, but they're all present in the same tube. The variant has a small domain attached to it that captures a stable RNA whose sequence identifies the variant of the protein of interest. The degree of RNA capture by each variant depends on the degree of folding of the variant relative to that of wild type. We take all of the we capture all of the variants, we convert all of the RNA barcodes into DNA, and we sequence them by next generation sequencing. So this is kind of like proteomics. We're measuring the abundance of every variant in the sample, let's say relative to wild type in one experiment, but we do it by genomics because proteomics is hard. Now, let me show you that it works. So here is BRAF. It's the protein we started on first because my colleague who helped develop Voodoo is a protein kinase guy. We made every possible missense mutation of this kinase. At every position, we have all 20 amino acids in the same tube. And in blue, dark blue, you have the guys that are underpresent relative to wild type. And white would be variants that are expressed similar to wild type, and then orange slightly higher than wild type. You get a few variants that actually overexpress the protein, but they're rare. Okay, so that's fine. So we give you the Voodoo score for these 15,000 missense mutations in one go. Now let me prove to you that it works. So the stripe here, the horizontal blue stripe, is the stop codon. We add a stop codon at every position along with the other amino acids. Okay. And of course, if the protein goes along and is truncated in the middle, it's not going to fold and it's going to be degraded. In fact, virtually all of them do, except when you terminate them at the very C terminus, the protein, which is mostly full length. It's missing a few residues at the end, still survives. Here's a hotspot. If you change this residue to most of the other amino acids, you lose the protein. Now, the real proof, we took some of these variants, selected randomly, and we made them individually in cells, and we measured the amount of protein by Western blot, okay? One by one for these variants. And you can see the Western blot level of the protein versus the Voodoo score correlates perfectly. So this works. Okay. Now it's time to turn Voodoo loose on all monogenic diseases in newborn sequencing pilots, focusing on those for which we don't have biochemical screens on the horizon, and for those in which a biochemical, well, I already said, our initial list just got to get started, pyridoxine-dependent epilepsy, Alström syndrome, high fructose intolerance, glycogen storage disease type 1A and 3, Leber congenital amaurosis, hypophosphatasia. We think that Voodoo has the potential to be applied to hundreds of these actionable pediatric diseases included in newborn sequencing panels. We think it can be done in less than 1 year by a single lab. Voodoo was developed mostly by Jessica Simon, a graduate student in Dusty Miley's lab, and also help from Yushan Liu and my lab. Together, we're working on Voodoo. The MLD ARSA genotype phenotype, my lab and BioMarin I mentioned, and the multiplex developed by this team in my lab. So thank you very much.